Vamos a comenzar el seminario. Buenas tardes a todos. Buenos días, buenas tardes. Buenos días. Gracias de nuevo por venir. Hoy vamos a continuar la serie de seminarios de RCMI y es un tema de mucho interés para el desarrollo de bioinformática en el recinto. Tenemos una vez más invitados a Carlos Rodríguez y a Humberto Ortiz del High Performance Computing Facility. Hace unos meses atrás ellos estuvieron presentándonos un seminario sobre Bionopics y hoy van a estar dando una presentación sobre Emboss, una herramienta computacional para análisis de secuencias. Así que les agradecemos mucho la presencia a ambos y esperamos que sea del agrado de todos. And we're going to start in English now. I just forgot, so I started speaking Spanish. So this seminar will be given by Carlos Rodriguez and Humberto Ortiz from High Performance Computing Facility. And it will be on Emboss, a tool for the analysis of DNA sequences. Thank you, and go ahead.
Hi, good afternoon. So today we are going to talk a little bit about Emboss and WMboss. Emboss is, okay, let's begin of who we are. Okay, we work for the High Performance Computing Facility. This is a UPR facility. We develop technology and give services to the UPR community. And now we are giving services to academic community as well, not only UPR. We are involved with the Inter and other institutions besides UPR. So my name is Carlos Rodriguez. I'm a computer engineer. I have a certificate in bioinformatics from Canada Bioinformatics Workshop Series. Humberto, most of you knows Humberto. He's a programmer, archaeologist. Ask him. He's a bioinformatics guru. You can ask him about anything in bioinformatics. He will answer you. And Daniel Ayala is working with us at the HPCF, and he is also a candidate for the bioinformatics certificate. So, as part of our work, we give services to the UPR community, such as internet to connectivity, software development, scientific computing, which is what we are going to show you today, databases, web services like web pages, email, mirrors, etc., video conferencing, and training and support. So what is Emboss? Emboss is the European Molecular Biology Open Software Suite. This is an open source software for molecular biologists. And it's also a framework for programming. You can develop your programs to work seamlessly with Emboss. You know, you can develop a software and make it looks like the whole package, you know, being consistent with what Emboss is. So what is Emboss good for? So as you can see, we have different categories here. It's more than 100 programs that cover sequence alignment, database searching, protein motif identification, nucleotide sequence pattern analysis, codon usage analysis, sequence pattern identifications, presentation tools like graphics and stuff like that, and other stuff that you will see in a while. So a little bit of history of Emboss. GCG, which is the Genetic Computer Group, this is an institution from the University of Wisconsin. They developed what was called the Wisconsin package. And they made the libraries and all their software available to the community. But like many things happened, this institution became private, and they stopped giving away their source code. So by the time they went private, many researchers already had libraries and software that they have developed before, and it was called eGCG, like Enhanced GCG. And so these developers created like a new alliance between them, and then they created Emboss as a substitution of GCG, like an open source substitution of GCG. So what are the pros and cons of Emboss? So as I told you, it's free. You don't have to pay anything. And it's free as you can do with it whatever you want. You can redistribute, you can modify it, you can sell it, you can do whatever you want with it. It's a replacement of GCG. Runs on practically every Unix, like Linux, Unix, HP Unix, whatever Unix, and Mac OS. Handles most of the sequence formats, integrates other available package, like BLAST, for example, and encourage developer to use the Emboss libraries, like for adding software to the package, not just doing another software. And the negative part is that there's no native Windows port. So like, actually that's not true at all. There's no supported port for Windows, but there are ports. You know, people have compiled the software for Windows, but it's not supported by the Emboss organization. Okay, and another thing that it's a bad side for many people, but it's good for many people like me, is that it is a command line syntax. You know, you have to write everything you want on a terminal, and many people find that awkward that you have to do that in 2005, right? But that's very powerful for people that don't want to be on a graphical interface all the time. So if people want to automatize work, that's a good way to do it, having a command line program. So given this fact that Emboss is a command line package, there has been many people develop graphical interface, like JEmboss, which is the officially supported graphical interface. This is a Java program, and it's very similar to what we are going to show you today. Also, we have web interface like the one we are going to show you today, WEmboss. And these are the most popular because you don't have to have anything on your local computer. You just have to need the Internet Explorer or you know, a web browser. That's it. That's the only requirement for the user to have. So WEmboss is what we decide to go with. Some of the advantages of this package is that each user has a separate workspace. You know, each user individually has a workspace where they will have all the analysis that they have done, it will be stored there. And you can organize your work by projects and sub-projects. The results are saved. You have help of how the programs work right there. You don't have to look through the web or whatever. And you can search for programs and documentation, and it will notify you if your job is finished. If it is a long job, it will notify you. So now I'm going to let Humberto explain a little more about Emboss. He's going to do the demo.
¿Se prendió otra vez? No. Ahora. Okay. So I'm going to go back so that they can see what we're doing.
Just close
Este window. No me deja. No, tampoco me deja. Sí, tampoco me deja. ¿Ve? I'll go in. Es que como tiene muchos frames, no de eso. Sí. So the address on your browser is wmbos.hpcf.upr.edu/little big EMBOSS. Okay? Or if you have any trouble, you can contact either Carlos or me. We need to set you up with an account before you can use it anyway. As Carlos said, each user has a space to store the results, and that needs to be set up beforehand. Okay, so I'm going to go ahead and log in. Yay. And I'm going to just talk briefly about the interface that you get. Emboss is different than most other web-based sequence analysis tools in that its main focus is around files that you store on the server. Most places you just cut and paste your sequence in and send them off, right? Which is nice because you can do it really fast, but it's bad in that if you want to use that sequence again for something else, then you have to save it on your computer and then figure out where it was and then paste it in again. Whereas in Emboss, the whole idea is that you store all your sequences on the server, and then you have immediate access to them from anywhere. So I'm going to go ahead and make a new project to store the sequences that I'm going to be using today, and I'll call it RCMI. And then I'm now in the RCMI project, and I have no sequences, no project files except for a list, empty lists of proteins and nucleotides. I have no program results because I haven't done anything yet. What I do have is a list of program categories. Voy a tratar de que este. Of program categories and search box for searching for programs. Okay? Like Carlos says, there's over 100 programs, and you can actually get a listing of all of them alphabetically here. It's a lot. Okay? So I'm going to be running through some examples. The nice thing about Emboss is that the whole suite has documentation available with the programs, and that you can search through them. So, for example, one of the one of the programs is called WASP name. Find programs by keywords in their online documentation. Okay? So if I say I want to know what programs can I use to analyze protein sequences, I'll put protein there and hit run WASP name. It'll give me a new box with a list. Okay? Find antigenic sites in proteins, back translate a protein sequence, do a charge plot of proteins, report stop codons, etc. And the same sort of output I can also do in Emboss by searching here, protein. Okay. And I'll get 150 matches found. And I'll get a link to the program and a link to the documentation. Okay. So if I want to figure out what Tranoline does, I can go straight to the manual and get the full documentation for this program. Examples, command line arguments, usage. The different options, the file formats that it can read, and output file formats that it'll produce, and you'll even get the references for the programs if they've been published in the literature. Okay? So the documentation's really complete. It's the best place to go if you need any help with any of the programs. So now that we've got a base, right, for finding programs and searching them, let's take a look at some of the programs that we have available. Okay, I'm going to go in and grab some sequences. So for example, the program for retrieving sequences is called secret. Reads and writes sequences. Okay? This program can use databases that have been stored on the server from Emboss databases. From my computer, I can upload sequences, or I can give it names of sequences that I've put in the sequence selector. So I'm going to start grabbing something from a database. I'll use there's a tutorial that comes packaged with Emboss, and it has a set of databases packaged with it also that you can go through and work with. And one of the sequences there is frog rhodopsin. If I run this analysis, I'll get a sequence. Xenopus laevis, rhodopsin, mRNA, complete CDs, right? And here's the accession number. I can save this onto this computer by clicking here, but if I go to my project, it's already been saved on the server. So here's a FASTA format sequence that's the result of that last analysis that I did. And if I want, I can copy this into my RCMI project so that I can continue working with it. And now I have it available here in my project files. Okay. So, and I can edit that sequence if I need to, make some changes. Qué chiquitito está esto. And work on it. So if now I have a sequence. That's good. Now I want to do some more analysis on this. So I know, for example, that I can compare pairs of sequences. I'm going to use a program called dot up. Okay. That displays a dot plot of two sequences. So what two sequences? Well, I've already got XL roll-up pasta on, no, este. No, I can use it from here. XL roll-up dot fasta. And then for the second one, I can grab it from the database. Yo quiero XL 23808. And then I can run dot up. And somewhere I should have results. Okay, open Apple tab. Oh. ¿Qué lo hizo? ¿Qué lo hizo? No me dio. ¿Corrió o no corrió? Oh, que no le gustó mi secuencia. Así. I'll just do it the hard way. Ahora. Whee! So XL 23808 is the genomic sequence for XL roll-up, and you can see that there's 5 coding regions in this. And that. And they show up quite nicely in the dot plot. So you can compare any pair of sequences like this and this one. In the project, we can go about this also another way. We have a nucleotide list, which I can edit. And in this nucleotide list, I can say I want to analyze XL roll-up FASTA, which is the sequence that I have here. And from the database, I want to analyze XL 23808. And I'll save this as my nucleotide list. And now I can choose those sequences from the sequence selector. Okay. So now my project file has 2 sequences, XL roll-up FASTA and Emboss XL 23808. ¿Que no le gusta esa secuencia? Wow. Let's edit it again. Quizás no le gusta el file. It really doesn't like that sequence. Okay. Yeah, ahora está eso. So if you've got a set of sequences that you use frequently, you can save them into this nucleotide list or protein list, and then you can operate on them through any of the programs by simply selecting them from the list. like I said before, if you've got some sequence here, you can also upload it into the analysis routines and then save it for further analysis. That's probably the easiest thing to do. Y ahora. Présteme un... I'm going to peek at the tutorial because I forgot what goes next. ¿Tengo internet? ¿Estaba conectado?
Sí, tiene un tab, lo que pasa es que no tiene el browser. No tienes la...
Oh, gracias. ¿Cuál más? Se supone que estén. View, toolbars. Ese es el que quiero. Dámelo, por favor. Me escondiste los toolbars. Me escondiste los toolbars. Open location. Jajaja. Tampoco. Carlos, me rompiste el browser. Ah, gracias. Trampa. Show DB, secret, infosec. Infosec lo voy a brincar. Dice dot plots, alineamiento. Vamos a hacer unos alineamientos.
Manzanito W.
Para otro de eso.
Manzanito W.
¡Qué! Gracias. So, ¿qué dije? Needle. So, quiero hacer sorry. I want to do sequence alignments, and I know a couple of programs. I'm going to use Needle. There's a global alignment of two sequences. And I can choose, let's see, XL Rodop and XL 23808. And then run this alignment program. And I'll get a multiple sequence alignment, a pairwise sequence alignment, a global sequence alignment. And if I scroll through, I'll have parts of XL 23808 and no XL Rodop. And then a little further down, I'll start seeing stuff like this. A coding region. Una, dos, tres, cuatro, cinco. Qué sorpresa. So we can again see that we've got a perfect alignment of the coding regions with the genomic DNA in these 5 exons. ¿Verdad? So we're happy. And again, if we go to the project, each of the programs that we've run produces output that's saved on the server so that we could go back tomorrow and review these results, okay, without having to run the analysis again. Okay, or I want to print a copy of that dot plot that I did for our lab meeting, so I can choose it from here.
No, just click that.
Acá. And get it again. Okay. Or copy them into my results, or go do some other work. Okay. So, ¿qué más?
Humberto, that's cool, but
But if I don't know
The name of the program that you want to do?
of the sequence, that, for example, I know the program that I would like to look for GST1.
Okay.
And,
How would you go about finding one?
yeah, there's several of them. And then,
The easiest way to do it is get the accession number from GenBank,
okay,
and then you can query it
over here.
over here. We actually have, I'll do show DB. So does anybody have a favorite accession number? There's a program called ShowDB that tells you what databases we have available. And we've got basically all of GenBank. There's a local copy at HPCF. There's a remote copy through SRS that's completely up to date. That's maintained by Indiana University. Again, GenBank via SRS, EMBL from Sanger. And you can query all of them for any sequence. You can use these names before the colon and get any accession number. So, ¿alguien tiene un accession number? Nadie. We could go search for one. Ay, no. XL Rodop funciona en Excel 23808. Dime, ¿cuál? ¿De dónde? Lo más seguro. No, ese es el número de ese. No, yo quiero un accession number, sí. O de SwissProt o lo que sea. Sí. Anyway, mira, vamos a hacerlo. Vamos en CBI. Dame. Aquí, ¿qué? ¿Cuál es el...? Window. Y aquí puedo abrir tabs. ¿Me deja? Y no se rompe. File, new tab. www.ncbi.nlm.nih.gov. So, tú quieres nucleótido o proteína. ¿Cuál era? GST. Voy a encontrar un montón.
Sí,
Human. Dale. Lo buscamos así y después le decimos que es human. Xenopus laevis. Yeast. Salmonella. ¿Solamente 2000 y pico? A ver si me deja hacer human.
Coge cualquiera en realidad.
No importa. So aquí tengo uno. ¿Verdad? Tengo el accession number. Sé que está en GenBank. So here's an accession number. I know this is in GenBank, so I can go back to Emboss, maybe, and do a secret. I want a database. I know it's GenBank. ¿Y qué? Sí, pero le tenía que decir. Le tenía que decir primero. Uy, ese no. Ay, sí, ¿te acordaste? Open Apple C. Whatever. Edit copy. No, control no es una Macintosh. Open Apple C, ¿ves? Sí. ¿Me va a dar? ¿Está pensando a ver? Hello. Contesta.
¿Dónde era que estaba ese servidor?
¿En el HPCF? Sí, pero no le he dicho nada todavía.
Está buscándolo ya Sí, pero está buscándolas todas. Ya, eventualmente te va a contestar que no puede.
Que no sabe qué va a hacer. Si yo le di ron. ¿Tú me viste darle ron? Yo no le di ron, ¿verdad que no?
Le diste tab.
Le di tab. Ay, no puedo. Escape. Vamos a eso. Vamos de nuevo. Secret. Vacío, gracias. Emboss. Genbank. Open Apple V. Ahora le doy ron. Él le estaba preguntando a ver si había alguno de esos allí. Y ahí tengo la secuencia. Okay. Y está guardado en el servidor, so you can keep on doing any kind of analysis. Okay. So yes. And we've got local copies of most of these databases that are relatively recent. They're maybe a couple weeks old. Okay. If you need any particular release of the databases, just let us know, and we'll set it up for you. So, tutorial. No, it's gone, ese mismo. All right. So local alignment's the same. I'm going to skip it. Protein analysis. We can do a plot orf. Okay. So tengo so I want to find open reading frames. I wonder what program I can use to do that. Ooh, look, I can do plotorf or getorf to find open reading frames. So let's do plotorf. Let's see what happens. I'm going to pick a sequence and then run it. I should have used GST. I like this one. So here's a plot of the open reading frames on the 3 forward strands. There's a nice big chunk. It looks like that's actually a real reading frame, right? It starts from about, I don't know, 100 and something and goes past 1,000. And nothing else looks like a real sequence. This is cDNA, right? So that's probably it. And then I can go back and say, well, let's find this. And this. Let's go through the list and see what we can find. So, ¿cuál era el otro? Getorf. EFG. Okay, I'm going to use a sequence selector to pick a sequence. And then I want nucleotide sequences between start and stop codons. See if I can find out exactly where that open reading frame is. Okay, and I'll do run getorf, and I get a nice sequence here that goes from 110 to 1,171. Yes, of my open reading frame. Okay? And you get the sequence, you get the start, you get the stop. Estamos bastante contentos. I like it. ¿Qué más? We can translate it back. We can have a look at secondary structure, right? Nos atrevemos. Vamos a ver el PEP info. So quiero, I want to have a look at the secondary structure. Let's go. Let's go grab PEP info. So I'm going to give it a slice. ¿Por qué brinca? Tengo que translate primero. Vamos para atrás. No. Puedo cerrar este. Go away. El transect. All right, so I've got my nucleotide sequence. And I want to translate the region from 110 to 1174. Now put the fast day and run. All right. 1173. Ahí está el stop. Se me pasó. But that's the sequence that I want, and it's on my machine already. Window, load the Emboss. I'm going to copy this file into my project. And then I can put it in my protein list. xlrodop.pep. ¿Qué será? ¿Que no le gusta este editor?
Firefox.
Que no le gusta el Firefox. Oh, va a ser un problema.
De Mac.
De la Mac. Si acaso lo abro en Safari.
O baja el file a la computadora.
Lo guardo acá y lo vuelvo a subir, sí. Let's try. Let's see if PEP info lets me. From the sequence selector, I want xlrodop.pep. Yay, it worked. And no. Es que le di uno de más. I'll fix it. There's one, the stop codon's in there. It didn't like it. I went one over. Project. Quiero este, and I want to edit it. And save. PEP info. So there we go. We got a hydropathy plot. Puedo tirarlo de aquí, ¿verdad? Dime cuándo. Ahí. Okay, so. And a bunch of auxiliary information about it, incluyendo down here somewhere way at the bottom, a nice plot of some of the chemical and structural properties of this protein. So tiny residues, small residues, aliphatic residues, aromatic residues, nonpolar residues, polar residues, charged residues, positive residues, okay? Negative residues. So tiene todas las propiedades de los péptidos. And in a nice plot. Y acá exactamente the actual values. In case you need them. So, okay, so we know a little bit about the information. We can also use a little bit about the structure. We can also use Garnier. If I take a piece of this protein, run Garnier, and it classifies each residue as helix, sheet, or turns, or coils. Okay. It's a little bit more secondary structure prediction. So you can't do 3D structure prediction so it's only sequence-based, but you can get some information about what's going on. And in fact, we can get some more. There's some nice chunks in this protein that were really hydrophobic, so you can ask what happens. Wonder if this is an actual transmembrane spanning protein. So we can grab XLSO up and do a T-map analysis, and it says, yeah, there's about 7 transmembrane sequences in this protein. Y te los presenta ahí. Right? And it actually is, right? This rhodopsin does sit in the membrane. So tiene an extraordinary number of programs for doing all kinds of different things. The hardest part is finding the programs. The tutorial helps. The search for keywords helps. And looking through the output of WOSS name also helps. You can also go through, let's see, okay, the categories to find programs that do enzyme kinetics, right? Or hidden Markov models for doing sequence analysis. For analyzing nucleotide structures for 2D structure, composition, CPG islands, finding genes. Okay. So you can go through the menus and maybe get some more information about what all the programs do. I guess the real secret is to get in and start doing some analysis. Yeah, so if you have any trouble, you can contact us, Carlos or me or Daniel at the HPCF, and we'll try to track down a program that can help you do whatever you need. And if it's not in Emboss, we can perhaps locate another program similar to that one in the open source community. Si hay alguna otra pregunta.
Are there any websites to open an account? Just have to contact you, and if you're a graduate student, we can open the account.
Yeah.
Okay, so we contact us by email.
Sí, Carlos o Humberto at HPCF, and then we can work it out. We have a account request form that's like 6 pages long. And we need like your social security number and your firstborn children's names and 8 signatures, pero we'll fax that to you. Sure.
Is there any way that you can get that information on the protein structure and actually grab it and use it somewhere so that you can actually have some sort of visualization of the protein?
Not 3D, not in Emboss as of yet. That's a really hard problem. I don't know of any good tool for starting with a sequence, generate 3D structures.
I'm just thinking, you'll have all that information about the residues and all those numbers.
Right.
Just thought about the possibility of just grabbing the numbers and getting at least some draft visualization of the structure, because the numbers are there.
See, a lot of them are. I for sure there's not any in Emboss. There's some sites that you can give them a protein structure and they'll maybe mail you back. They can give them a protein sequence and they'll maybe mail you back a structure. But it takes a lot of computational resources and it's not easy to do. So in general, you can't. So,
how about generating a figure from the output that you see?
Actually, you can just save these directly. If you, let's go back to our project. I've got my data results, for example. Okay, this is actually a PNG that you can save, right, and then import into Word or whatever. While you're doing the run, most of these also let you output to another format. I think it's mainly no. Let's run it again. Oh, come on. Adopté. If I pick my 2 favorite proteins, my 2 favorite nucleotides, and I can choose which output format here. So my choices are PNG or PostScript. PostScript you could import into like Adobe Illustrator and then annotate it and then export it to whatever. Or the PNG file you can also edit directly. You'll get slightly better resolution if you do export it as PostScript. You just need a different tool to mark it up. ¿Lo puedo ver aquí? What'll happen?
Yeah.
What'll happen if I click to view?
It will ask you.
Do you have Ghost View?
No.
No.
Do Adobe.
Adobe won't work. I don't think so. Adobe abre PostScript. I don't think so. Let's try. Lo voy a dar choose y darle Acrobat a ver si es verdad. Sí, eso es. Preview. No, Adobe. Entonces la Mac lo abre. El display PostScript, ¿verdad? ¿Por qué entonces no se da cuenta? Tanto nadar para morir en la orilla. No de eso.
Pero es verdad, si lo guardamos,
Si lo guardo, si lo abre. Ya va. Save to disk. Ok. Ah, es que no sabe lo que es. Le tengo que decir que es PS. Punto PS. ¿Y ahora qué? ¿Eh?
¿dónde lo grabas?
Donde él decía. ¿Qué le digo? Ah, lo tengo que encontrar. No.
Download from
Catch.ps. Vamos a ver si lo encuentra. Enter no funciona.
Ah, ya lo está buscando.
Oh, perdón. Pero los PNGs como quiera los puedes editar. Y hay formas de editar el PostScript también, si acaso necesitaran. Ahí está, lo encontró, ¿no?
A ver.
Pero eso solamente en la Macintosh. En las PC tiene que hacer otra cosa. Pero vienen también. El tutorial de Emboss pelado, el tutorial de Emboss pelado es bien bueno, que tiene muchos de los programas que se utilizan en Emboss y te da ejemplos de cómo irlos corriendo y se puede correr completo en Emboss como lo estamos haciendo ahora. Vale la pena. El tutorial está en el site de Emboss, si se pierden nos pegan un grito.
Compatibilidad con browsers.
Anything mainstream will
Funciona.
work.
En algunos funcionan mejores que en otros.
Mhm.
Pero Explorer funciona y en la Mac funciona Firefox.
Safari doesn't work quite well because of CSS, but on the Mac, the browsers are not good.
on
So
the-
yeah,
You use a Linux or-
yeah. Basically, if you have a PC, you're okay. If you use Linux, you will use Firefox or whatever, and it will work. And on a Windows PC, you can use Netscape, you can use Internet Explorer, you can use whatever one Firefox, and it will work. On the Mac,
A veces sí, a veces no.
so another question.